Thanks for this opportunity to talk. I really appreciate that. And I can't see Q&A right now, but I think Daniel can. I can see your chat, and I don't really mind if there's not even a huge crowd here. So I think it would be fine if you guys throw any questions you have in the chat, and I can try and answer them live. That makes it a lot more interactive if you have questions. Yeah, so I just want to introduce my company and what we're trying to do with Radical and TypeDB, and why we're doing it, and then introduce you to the virus graph as well, which is just one of many things that we're doing that are along the same lines. So yeah, that was a great introduction, Daniel. I wanted to say I've been involved with Radical and Kraken for a number of years, probably 4 to 5 years as well. And it's been just a really supportive community. I think you probably all know this if you're here, but this company is really cares about knowledge graphs, right? Really cares about utilizing knowledge graphs and information and democratizing the information by making it accessible, like a very complicated structure, making it accessible in human terms, right? So let's jump in with that, and I'll just show you what I've got here. I presented this two months ago, and hopefully it's increased. It's changed a bit, and we've gotten some more maturity on the project. This is my company, Austin Capital Data, and it's sort of hand in glove with the mission of a group like TypeDB. We're very much interested in engaging with the community. This is the Austin bat swarm that comes off of the bridge. There are millions of bats that live under this Congress bridge, and my wife suggested that this would be a good logo for the company because it's a structure that has complicated dynamics. You know, each of these bats has to look at its position in space, and they all are taking in information as essentially nodes on a graph, right? And then the thing that emerges is this swarm, which is a funnel that does good service, right? And the good service is, you know, eating bugs that are trying to eat our corn. But anyway, so yeah, this is me. I'm Gunnar Kleemann. I am I'm a biologist by training, but I'm very interested in pushing forward these domains and making them accessible to everybody, right? So that's the main mission. So let's jump into that just a little bit more. I'm also so I was trained as a PhD biologist. I did that for what, 15 years. Went to postdoc and studied genomics. All incredible experiences, but that information is very privileged, right? Took me a long time to understand it. It's very hard for me to even explain it to somebody who's not initiated, right? And there are very few people that are initiated. This runs along the lines with data science as well. The issue is the same. Much of today's important data is hard to access, utilize, and derive insight from. Now, this isn't because it doesn't belong to us, right? This information is actually all of ours. A lot of us have paid for this, especially if you're paying in, in England or anywhere where we've got active research programs, you're all paying taxes that go into building out these data structures and this information. There are a number of challenges that actually get in the way of us understanding information. Data management is a huge deal, right? If it's like Twitter data, you've got, or paper data, you've got to do actual NLP on it. If it's a raw data stream, it's an issue. And then even the biological data that I'll talk about today is also relatively difficult to handle, just because of the large structures that you have to deal with. Then there's another level, there's data analysis, right? So how do you derive insight? Once you've cleaned it, I think a lot of people talk about this, yeah, once you clean it, it's easy, the insight comes without any effort. This is 80%. That's true, but it's not really true. Because once you clean the information, what do you do with it? I don't know if you guys have experienced this, but maybe you've spent a lot of effort cleaning information, and then you find that it's really hard to get at something that's novel and actually resonates. Knowledge graphs, this is the reason why we focus on knowledge graphs and we engage so closely with Radical. Knowledge graphs help bridge this gap, right? This is one of the best mathematical structures that allows us to simultaneously look at all this information. So that's the company vision. We have a mission of educating and enabling with data. So all of our projects are focused on this in various ways. I pick enthusiastic people from the MIDS program, the Berkeley MIDS program, people who are the best talent that I can find, and we bring them onto these projects. So that's enabling them to move forward. I also, all of our projects are based on like taking complex data and putting in a usable format. So hopefully this talk will do that. And then the other thing is we want to find problems, right? And try and meet new problems with these new tools, right? So yeah, one of the biggest upcoming challenges, now that we've got the data science revolution, right? We understand how to handle large data. It's sort of commoditized where you can actually pipe stuff in. They're acceptable practices, data lakes, right? But the biggest challenge is making this useful, right? That's the next challenge. So hopefully I'll make a step forward on that. Okay, so here are some of our data stories. The first one we had was BioPredictor. So this was a time when me and the people in my company were looking for jobs and we wanted to know where to go, right? So this actually sort of a little bit predates the company, but it was a group of interested people, right? And it turns out we were all biologists and we knew how to pull data from the web. We know how to parse data. We know how to understand data. So what we did was we built a machine learning model that pulls all easily accessible biotech companies and was trying to predict which ones are going to be successful, which ones are going to IPO, right? So we were able to get relatively high prediction using, it was logistic regression was a pretty good way to predict whether a company was going to IPO or not. So that was our first project. And then the rest of these projects, so this is a basic machine learning project. The rest of these projects are like collaboration graph. We are very interested in organizing scientific knowledge, and you can pull PubMed data, right? But one thing that's not easily accessible is who's talking to who, right? So collaboration graph was this, and I was able to pull my PI from postdoc, my PI from graduate school, my PI from undergraduate, and compare how these people work with other people. This is really interesting because like, how do you judge a scientist? You usually judge by the number of publications and where they publish. But I would argue, right, that it's more important how these people work with other people and what their friends know, right? To really judge the quality of that scientist. Are they good? Are they working with people who know what they're doing? Are they very collaborative? Are they not? And I guess I wouldn't drop any judgments on that, but I would say that those are qualities that should be measured. Okay, I'm going to talk about Cancer Graph at the end. But Cancer Graph is actually a personal story. It really hits home in terms of what I think we want to do. My sister had cancer, and I asked her, if we could build any data science tool for you, what would it be? And she told me about all the missions of Cancer Graph, which is basically allowing somebody with cancer to take their cancer subtype and dig into the graph and dig into the literature and find out, who's doing who's making new treatments, where's the best level of success, where are the centers for, it was pancreatic cancer with her, what are the world centers for that? Not so easy to get that from the guy down the block, which is what a lot of people would do if they had cancer, right? You have your doctor, and then for other information, you go to your circle of friends and try and figure out if there's anybody who knows about this stuff. Well, the idea is to put the cancer literature into people's hands. Cocktail Graph is a postdoc of mine. I said, look, do whatever you want. She was a bartender, and she said, okay, well, I'd like to make a cocktail graph. And basically look at all cocktails and how they relate. I said, okay, great. Now, what we're going to talk about now, Education Graph is another one of my people. So basically, Skylar is homeschooling her kids, and she built Education Graph. And the idea was that the homeschooling curriculums are very confusing, right? So of course, you can ask people what they like, but we want to put the actual curriculum data in people's hands if they're so they can make informed decisions. And then finally, I know I've sort of waxed poetic, as Daniel was saying. It's hard not to. I'll land on Virus Graph, which is our newest graph. And actually, Sandeep, I'll mention him again, but Sandeep, who's here, is working on building this out. So The idea behind virus graph is there's sort of an opportunity, right? So we've been working with BioGraphing for quite a bit, which was essentially donated by Vatical, and Tomas at Vatical built this, and there's probably other people that participated, but I know he did, and it's a biological graph, but it's also there's the aspect that they looked at 15 viruses, right? So they actually added data from 15 viruses, all relatives of COVID. So this is an incredible opportunity to just think about viruses in general and how you can use graph analytics on viruses to understand them better, right? I'll tell you more about this as we move through. But the idea is that we have pandemics, and I think people are aware of this now. One stunning data point that I think it's worth mentioning is that Obama, made a pandemic committee, right? He built a pandemic committee. And the first thing that Trump did, I don't know if it's the first thing exactly, but he disassembled it, right? When he came in, we don't need to talk about pandemics, right? And obviously that was an odd decision at that time. And it turns out it actually probably wasn't the right one. So it's a nice, interesting data point. So my point is that there was a difference in opinion whether epidemiology was important, right? Maybe it's not even important. Maybe we shouldn't spend that money. Well, I think now, we understand that understanding viruses is actually a good cause. So the question is, how do you understand virus biology? And can we do a better job of understanding virus biology as a culture now, right? There's a priming that we've been through. And then maybe we can predict new pandemic sources. Maybe we can build systems for threat monitoring. Those are open questions, but I'm going to bring up some ideas and just sort of drop them in the collective space and see if they start any fires. That's the idea here. Okay. So again, pandemics are here to stay. You can see that we had smallpox for 3,000 years with 300 million deaths, right? That's obviously a problem, right? And it went all the way till 1979, right? That's actually modern history. Black Death is probably the most compelling situation, right? That was in 1347 to 1351, 225 million people in much smaller span of time, right? I mean, horrific, right? And then there's Spanish flu, which actually killed my great grandparents. So that's 50 million people, right? And not good at all, right? And then HIV, we know about HIV and AIDS. This is another really interesting case because whereas I don't know how many of you remember all the politics around this, but quite interesting about the politics of HIV. Obviously, it was a lot longer period of time than like COVID, but you can see that there's by looking at like comparing Spanish flu, Black Death, and HIV, you can see that there's a layer of opinion about these viruses based on the presentation, I guess you might say, and that's something that I think we can tease apart with a graph analysis. Okay, cholera, right? Asian flu, right? This is more recent. Russian flu, Hong Kong flu, swine flu. You probably remember this. I have friends that got sick with this. And obviously the numbers are not as stunning as before. COVID, this is obviously old, right? Somewhat dated, but we can see Ebola. These, were relatively contained, and then there's MERS and SARS, which were contained as well, regionally contained, right? So, yeah, that's the point. Is I don't think it's over, right? There's going to be more. We're going to add more to this list. It's just biology. Okay. So what do we do, right? Where do we go with this? I mean, we can basically, here's your viruses, this is COVID. We do research, right? Research is done on these things. We have that. That's sort of pipelined already. Like, if there's a virus, there's going to be, that's dangerous. There's going to be a research pipeline. But the question really is, at least the question for me is, who owns the knowledge, right? I'll come back to that question. Who owns the knowledge? Is it some brilliant guy who's standing looking at the horizon, right? That he's gonna, figure out the solution? Or is this really all of our responsibility to understand this, right? And I believe that the knowledge graph is actually part and parcel to this question. It's not so much whether there's intelligent people who are not engaged in this, right? There certainly are intelligent people who are not, who don't have access to this information, but it's more about like, what is the technology allowing us to do? You know, before, right, this person had to read, you know, hundreds and hundreds of papers, have a nuanced understanding of a very small drill down, right? Even in science, like if you're a biologist, I wouldn't understand COVID biology unless I spent a lot of time reading all the papers, right? And I'm not even going to argue that I'm an expert, even though I'm talking about this. What I am going to argue is that I believe that we have tools that can allow us to get deeper faster, right? So yeah, just that's the fundamental question. So here's the virus graph. What I wanted to do was just basically put this in a understandable, interesting structure, so that we can start to think about viruses, and maybe, you know, throw some ideas out there about, you know, what's important about thinking about viruses. Are there things that we do that we should augment, right? Are there ways of thinking about this that we should, you know, all adapt? So these are 15 COVID-related viruses and their proteins, right? This is straight out of BioGraken. We're adding more data, but we haven't done this for this talk. It's not all in there. This is just the stuff that's in BioGraken. So understanding viruses and the human proteins that they co-opt and attack. I want to, you know, just hang on that for just a second. This language, right? This language, co-opting and attacking. I think it's for me, it's coming from cyber threat analysis, right? Where you think of a virus as a potential predator for your computer, right? As a virus as a threat. I know we all think about viruses as threats, but if you look in the scientific literature, there's a lot of ontology. There's a lot of protein function. There's a lot of, you know, the first thing that comes off of a viper is this huge genetic sequence, right? Sequence alignment. It's all stuff that it's very important, but it's not, you know, the language of the first thing I want to think about is threat. Okay. So leverage BioGraken COVID, which is TypeDB. Let's think about the data that's available. So the data that's available is an annotated data set of coronaviruses and the potential drug targets. And that was put out by Oxford Biogenesis. It was donated to the cause by them. And it was based on a hand-curated literature review, right? COVID-19 relatives, there's 15 distinct gene bank numbers and 133 virus interacting proteins. So you can see all that data. You can download those two data frames. And they've got attributes as well, like where did they come from, what's their primary host, that kind of stuff. All right. We can see these in terms of their spread, in terms of their host, right? And then the discovery site. So these were human viruses. These are all found in China, and there's one avian virus. These are largely human viruses, but there's mouse and avian as well, viruses included. Some of these are found in the US, right? South Korea, Saudi Arabia. Obviously, looking at this, you know, this is nowhere near, it's a very small slice of viral biology, but it's focused, right? It's focused on our current problem. Okay, so how do you analyze this? One way to analyze this would be to focus on protein enrichment. Let's see what proteins are enriched in this interactant set, right? Let's see if we can figure out anything about viral biology by looking at this protein set, or maybe viral genes, but in this case, it's the proteins that are enriched in the targets from the literature. So if we look at this, we see this is standard Go term analysis, right? Gene ontology term analysis. And this is like what you see a lot. I've seen a lot of postdocs and grad students do Go term analysis. And I don't want to insult anybody, but it always seems to be something like this, something very general, right? And what I'm going to argue is this is fine, this is great, it's necessary, but I think for the purpose of making this understandable to the general public, we need something a little bit more compelling, a little bit more, with a few more hooks on it, right? And where do you get that? Well, you get that by enriching the attribute space of those objects, right? So thinking about what are the attributes that those objects have? What resonates with people? What's actually important to people about this viral biology? So I'll just read this to you. These are 54% cellular nitrogen compound metabolic processes, right? Okay, so metabolism. 50% of these are involved in biosynthesis. 50% of these are involved in transport, and 47% of these are involved in signal transduction. Okay. Now, I mean, these are very general, and I would challenge anybody who doesn't have a biology background, or even somebody who does have a biology background, to say something substantial from this list of Go terms. Um, Pretty much, you'll see this for a lot of sets of proteins. And again, I don't want to say that this is not important, but I think there's more that we can get out if we think about what's an actual hook, what's actually compelling. Okay, so let's start to think about this. What human proteins interact with these viruses? What's the distribution of them, right? So we have 7 viruses with protein data, 99 proteins with 1 connection. So these are private, and then 7 proteins with 2 or more connections, right? So these are actually, I mean, these may actually be shared across viruses, but in this small set, we see that there's many of these proteins. The majority of these proteins actually are just interacting with one virus, but these pop out, right? So ROA1 right here, ROA2, ZCRB1, which is the zinc finger transcription factor, ACE2, BTF3, transcription factor, elongation factor, and FGL2. So these are things that we might look at because one or more virus, well, more than one virus is using them. So that says that either they convergently evolved, but more likely they just didn't, the virus as it changed, it just didn't stop using that piece. So those are things that we might want to think about. Let's see. Yeah. So you can see these here. I've just pulled them out. Human proteins interacting with these viruses. And you can see the private ones would be these, right? Private, and then things that are common pieces are those ones over there. So next, yeah. So if we take a step back and see what we did, just in that one step, we went from like sort of a traditional analysis where we take all the virus proteins interactions and then look at the enrichment in this protein set. And we've moved into a graph structure now, because what I did was I took virus, protein, and virus protein, and then saw where those things were common. So I'm looking for things that are shared versus things that are private, right? And that I think allows us to start to think about the fundamental pieces in the human, right, that are targeted by more than one virus. They're either weak spots, they're potentially useful, right? Maybe these could be drug targets, but that type of enrichment, I think, adds an element of interest to this. You can look at the code for this, right? So I'm looking for match V1 is a virus, P is a protein, and then we've got this relationship, protein virus association, right? And then I've got V2 as a virus, and then protein virus association. So a very simple query on Kraken to get that relationship out and build a table with it. And then you can see the output over here. So we've got this virus has all of these associated proteins. So this is what SARS-CoV-2, that's SARS, right? That's COVID, yeah. And then it's got these shared ones, right? ACE2 is one of them, and then FGL2, you remember that from the other, shared with HCOV229E, which is another variant, and HCOV-NL63, which is another variant with this one over here. And then you can see this one has another associated protein, but this would be this is probably curation bias, right? This is a highly curated virus, and then we can see other related viruses that share pieces, right, of a human, as I guess call it target. Okay, so so it turns out that there are, if you look at the virus protein graph, even though there's 15 viruses, 6 of these are wind up in the virus protein graph that I made. And I'll just point out that, well, what they are, this SARS-CoV, GZ02, which is SARS-CoV-2 is the same thing. MHV, IBV, it's bronchial virus, HCOV-229E, one of the variants, NL63, and then MERS-CoV, right? So these are all different viruses. If you look, the blue spot over here is a virus, and then the red ones are the proteins that are associated, right? So you can see that these are all associated with this virus. Here's a private one right here, and here's SARS-CoV. These are the two names of COVID. They pretty much cover the whole graph, but then they have these viruses that are sort of on the outside. Now, I'll show you a close-up view over here. So this is MHV, right? You can see my MHV. It's got this one private protein, ACDB5 human protein right there. That's right there. That's that part of the graph. And then you can see the red highlighting with the immediate links for SARS-CoV to the other proteins. You see it doesn't track to that human protein. Okay. Now we can set these up on a wheel if we want to, just to see how much of the whole graph. So this is a mix of proteins and viruses. Each of these nodes is one entity, which could be a protein or a virus. And then you can see these bundled links are the links between these things. So so SARS-CoV-2 has a lot of links in here, pretty much covering the graph. I can do the same thing with the alias of it, and it's got somewhat different links, but it's pretty much covering the whole graph. This is MERS-CoV. You can see it's actually targeting a few less places, right? That this is extended a little bit less. And I think this is sort of curation bias, right? But you can see how this, like focusing on COVID, for example, we can get a lot of information, and then other, things that are a little more distant, we can get information about them. MHV actually targets quite a few of this, quite a bit of this graph as well, right? Which is interesting. IBV, right? A different subset. And then HCOV-229, which is kind of interesting. It seems to cover quite a bit less. Maybe it's well diverged. That's open question. Just what I want to say is, by just perusing these, right, we're already generating questions in our mind. And that's really the purpose of the graph, is that to structure the thinking, right, in a very complex place without reading 100 papers, and say, okay, why is HCOV-229E different? Is it different? Is it highly diverged? Is it maybe not likely to be like a COVID type of pandemic? What's different about that? And I don't know the answers to that, but the questions would stimulate me to read if I had, if I wanted to dig in that direction. So that's where I want to get people. Okay, and then again, another interesting one. We got HCOV NL63, and again, this doesn't address a whole lot of the graph, right? It's just this one small portion. You can even see this more graphically, right? IBV, SARS-2, these are sort of similar, and then we got 229, which is less, right? And then you can see NL63, which is less. What's going on there, right? Is it, are those shared connections? Are those the things that are sticking? Is it a specific pathway that's retained? What's falling off on this one, right? So, yeah. So let's just dig in to the shared co-opted proteins, and 3 of them really stuck out. ZCRB1 had the most sharing, right? Row 1 and row 2, they were both in the top list, right? And row 1 had 2 sharings. And row 2 had 2 sharings, and row 1 had 1. So it looks like this RNPA1, HRNPA2B1, maybe some important biology as well, right? And it turns out, I'm not going to dig into these right now, but it turns out that these are involved in microtubule organization. They're also involved in transport of HIV, genomic RNA out of the nucleus. So there is a viral connection, right? It's transport and microtubules probably pulling around those particles, right? Those RNA particles. So that would be very important for what viruses do. Basically, it's a very stripped down replicating machine, right? So RNA movement is really probably very key for them. let's dig into ZCRB1. That's a zinc finger CHC type RNA binding motif containing protein, probably something like a transcription factor. Zinc fingers usually hop on the protein and promote transcription. And the question that was compelling to me here was, is there any data, and what's the data look like for these CCHC transcription factor type proteins to be involved in viral biology? It really looks like this comes up a lot in this data set, so why? And then we can actually see there's been an experiment and a review, right? So this is actually a field. So the role of CCH type zinc finger proteins in human adenovirus infections, right? This is not the same protein, but it's the same type of protein, right? It's the same family. And what they've actually done was they've been able to take wild type, which is just your normal garden variety. And then this one is a mutant that doesn't have that protein, right? And you can see this zinc finger protein binding to NF kappa beta. This would be I guess it would be a normal function right here, right? And then NF kappa beta would promote transcription. In this case, this would be the viral late mRNA, right? So this is actually the mRNA, and the zinc finger is also, we hypothesize, binding to that and causing the replication. And what you can see, if I'll just focus on this, is that the wild type produces more late viral proteins, and the knockout produces quite a few less. And that would argue that this CCH type zinc finger protein is required for the promotion of the production of viral mRNA. So there you go. Now that process, although you could say, and most of my scientific colleagues would say, I don't care, it's known, right? There's a review. But I would say that our target domain is really not scientists, right? They are going to know this, but it's generating interest and understanding in the viral process, because I would say that, you know, now viral biology is not just the domain of virus experts. It should be the domain of everybody. Like, you might want to know how a virus works and what those specific targets are. So how do we make this more palatable? It turns out that there's a nice story to be told if we look into the world of security. Biological viruses are not the only virus that threatens us, right? We've also got cyber viruses. And cybersecurity people think in a very different way from biologists. And I would say for this application, this might be much more compelling. So the cyber threat community uses something called STixx 2.1 and other CTI tools. STixx is a classification of objects, a lot like, it's an ontology, right? It's a lot like our ontology, but it's only 18 items. And it's all focused on threats and classifying these different pieces as threats. The two I think that are the most compelling for biology would be a tool, a legitimate software that can be used by threat actors to perform attacks. You know, your directory replication structure, whatever's inside the computer that the software is going to co-opt. I mean, the virus is going to co-opt to replicate itself and make scary messages, whatever it's going to do. You know, your directory deletion tools, whatever it is, the way to get in, right? Your pipes into your computer. This is very similar to the transcription, you know, using a transcription factor to replicate your viral proteins. And then there's this vulnerability, right? A mistaken software that can be directly used by a hacker to gain access to a network. This resonates with me in terms of like a transport protein, right? Something that the virus can use to get through the cell wall, right? Something that's going to recognize the virus as something that should be inside the cell and take it in. Those are pieces that we actually have a good idea of what those pieces are from all of our biological work, right? We could actually do the same thing for viruses, like biological viruses. So you can see basically what one of the really compelling things is in the cyber threat community, they'll build these stories, right? And with this recognized language. So you've got targets, you've got a campaign. This campaign will be, as it's detected, will be streamed to all cyber threat analysts, right? Through a centralized system. The threat actors will be attributed, right? So you might say who's doing this, where is this coming from, what was the source of this, right? Is there any more source? And then there's the indicators, right? So these are things that actually we can use as bioindicators or indicators of a cyber virus if there's specific directories attacked in the case of cyber viruses, right? So there's that, and there's 18 of these. You can look at these on, there's a 200-page paper on this if you want to look at this. Looks like we're running low on time, so I'm gonna move quickly, but I think this is pretty compelling just because it's focused on attack, it's focused on defense, right? It's not focused on where something resides in the structure of all 60,000, 100,000 elements, that could make up a biological entity. It's much more focused. So we worked on this for a while. This is OpenCTI, right? So this is one of the tools that's used to suck in all of the current viral threats and historic viral threats. You can see how many entities are in this, right? So this is a brilliant tool that actually I think would be great to have for a biological application as well. Where are we going to go with this? We're building out the queries to look at virus to virus path, like I showed you, but with more in between, right? So protein, function, protein. So this would obviously be quite a bit bigger graph, but perhaps we know viruses are going to target certain parts of the functional ontology. So we're building that out. Sandeep is here actually, I think, and he is building out the all virus graph, right? So using Viper, he's actually currently, like as we're speaking, probably loading viruses into our graph. So we can understand, viruses in general. Maybe look at this, look at the vulnerability space. Like we know COVID was a problem. We know MRSA was a problem. SARS was a problem. We know are there other things that may be problems, right? Based on the traits and the characteristics. Could we apply a neural net on top of this to take known vulnerabilities and predict future vulnerabilities? These are really interesting. Cool. And then, so that's that. I should say a little bit about Cancer Graph, but I'm running really low on time. I think Cancer Graph is very relatable. It's a personal story. The reason why I bring this up a lot is because I think if you find yourself in this situation, you may want more information and be it'd be very hard to get that information, even if you're a biologist, even if you're a smart person, right? It's just a lot of information. You'll have to rely on, people like doctors, which is fine, but it's also nice if you can understand things yourself, right? Like, for example, for pancreatic cancer, there's 105,000 references. Where do you start, right? How do you develop an opinion? Cancer Graph is really, really lean, right? And the idea is that we have mesh terms, authors, journals, keywords, chemicals, and we're able to classify all papers and objects that way. Yeah, this was the structure that we built out to look at Cancer Graph. And then we found, because my sister was very interested in side effects, it's actually a huge problem when you have cancer, because you could actually choose to die versus taking your chemotherapy if it's really bad. And we found these two highly cited chemicals that are actually antagonistic to each other. So she could have dug into that, right? So there's that. Without running over, I'll just thank everybody who's involved on my team. Me, Rob, most of these people are coming in from Berkeley, except the people from Austin who have been, pulling the smartest people from Austin. Of course, they're the smartest, right? No, I've been finding very smart people around here, and then when I find somebody who I think could really fit in, I bring them in. So that's Skylar and Myra are people who are, who have come on board as well. Everybody else is Berkeley mids person. So, all right, yeah, we've got like 10 minutes left, so let me stop talking.
Well done, Gunnar, as always. Thank you for sharing that. So as I mentioned in the chat, feel free to raise your hand in Zoom, and I can bring you off mute to share thoughts, curiosities, questions for Gunnar, and we can dive in that way. I'm particularly curious, Gunnar, to sort of kick things off. Where do you see these projects going in, let's say, the near term, and what do you hope, like, for those that might want to poke around and maybe they are experiencing a similar thing with a friend or relative in terms of the cancer graph application, how can the community sort of get their hands on this?
Yeah, so right now they're sort of all brewing in the shop. But yeah, we do want to open them up to the community. I think the open source initiative for Vatical would be a really great place to start to release these things, just to have a better platform for them, and allow people, if like I said, somebody's got cancer, God forbid, but if they really want to, work on the project and improve it, that would be great as well. I think a lot of these are going to have an open source aspect, maybe on the application side, right? So like open source front end, maybe able to download the analysis, this kind of a thing. So yeah, contact me directly if you're interested, or come to our open source OTI, right? It's OTI meetings. So we do have this meeting regularly talking about these projects.
Yes, absolutely. And we're going to be, so the TypeDB OSI, or the TypeDB open source initiative, is a group and a program that we've founded, I want to say maybe a month and a half ago. And as Gunnar said, it's meant to provide an ecosystem for the community to not only contribute to the development of TypeDB Core, our open source community version of TypeDB, but then also be building tools and software around TypeDB so that everyone in the community can be moving faster, be utilizing each other's work to further accelerate or progress their own development. And we will be launching that officially out into the wild at an event in, I want to say, on July 28th. So that is where we'll sort of announce to the world the initial projects and sort of framework that we've been working together with a group of community members, which Gunnar is one of. So yes, that is it. We really do hope that you bring these projects into the OSI, Gunnar, and looking forward to having you there. For those that are still on, please do let us know if you've got any questions. We know that this can be a bit of heavy topic, so if it needs some digestion or you need to watch the talk again, this will be on YouTube following today, following the conclusion of the talk. And then we try and continue conversations in Discord as much as we can. So do encourage you to join in Discord as well. All right, so anyone have any other questions, comments, thoughts, feedback? Otherwise, we can send you off into the Great Wander. Is that did I get that right? The great outdoors, the great unknown. I don't think there's such a thing as a Great Wander.
I think it's the Great Blue Yonder, right? Isn't that like a Air Force thing? I think it's the Air Force thing.
Yeah, exactly. I was trying to combine things and that should have been combined.
That's cool. That's good.
All right. So with that, Gunnar, thank you so much for sharing. We're going to have you back, obviously, for the TypeDB OSI announcement, and then hopefully in August, September, we can bring you back in and hear some updates on the projects that you're working on. If you want to chat with Gunnar, reach out about working with him and his team, please feel free to reach out to him in Discord, or I would imagine LinkedIn is also fine, but we'll pass around some ways of getting in touch after we get done here.
Fantastic. Yeah, LinkedIn is the biggest community I have. So LinkedIn and Discord, obviously Discord's pretty personal, right? So I'm able to see that very easily. So
absolutely. So Gunnar, last parting words before we send them off.
Mm-hmm. Yeah, thanks for coming. I really appreciate everybody listening. I hope we can do more conversation about how we can use graphs to democratize knowledge.
Love it. Thank you so much, Gunnar. Really appreciate it. Everybody have a wonderful rest of your morning, afternoon, evening, wherever you're calling in from the world. We're just glad you're here hanging out with us, and we will see you all on Discord.